Okay, hello everybody, and we're going to try and complete our discussion about viruses by talking about a few human diseases caused by viruses. And remembering, of course, that there are actually hundreds of different diseases that can be caused by viruses. The way in which we acquire viruses is usually through our face, in actual fact, either through the mouth or most commonly by breathing in droplets or by touching mucous membranes and infecting our eyes or the mouth or whatever. And that's not the only way that we can acquire viruses. Viruses can also be transmitted by a vector. So, for example, something like West Nile virus, yellow fever, these things are transmitted by mosquitoes. And in addition, sometimes viruses can be transmitted by animal bites. Rabies, for example, is transmitted by animal bites. But by far the majority of human viral disease is acquired by aerosol droplets. And so it's unsurprising then that our discussion today is going to focus especially on respiratory diseases and on those which are acquired by aerosol droplets. So, if this will let me go. So the question then is, how is it that viruses actually cause disease? Why does a virus actually cause a dysfunction in the human body? The first reason is, of course, that viruses are obligatory parasites. They require a cell, a living cell, to act as a viral factory, to produce new viral particles. And in doing so, they co-opt the functions of the cell. They drastically affect the cellular function during that phase when the virus is being reproduced. They may remain silent for a long time. That is true. And there are viruses which remain completely dormant and never make their appearance as a disease. But mostly, any time that the virus is reproducing, it co-opts all the normal functions, and it may in fact destroy the DNA of the host cell. And ultimately, the dysfunction becomes so severe that the cell actually dies. By doing so, viruses end up disrupting organ function and all of our homeostasis. Of course, the virus doesn't think, the virus doesn't have intention, but the evolutionary pressure has been to develop methods to reproduce as efficiently as possible. And if that is at the cost of the host, then that is just too bad as far as the virus is concerned. The other way that viruses cause disease, and often extremely serious morbidity and mortality, is by exciting a very strong response of the immune system. And an over-response of the immune system actually is frequently fatal to the host. The last thing is that in damaging cells and in damaging organ function, viruses predispose people to secondary infection, especially secondary bacterial infections. And that, for example, a lower respiratory tract infection by a virus may predispose to a lethal pneumonia. And so there is a chain of effects that arise from a viral infection. So we're going to start off by talking about the typical kinds of viruses acquired through the respiratory tract. And of course, the commonest one is the common cold. Now, I've emphasized in this set of slides the differences between the common cold and influenza. There are two entirely different diseases caused by entirely different kinds of virus. In fact, the common cold has a set of symptoms. It has a syndrome of symptoms. And these usually entail, as you well know, sore throat, a runny nose, which is actually the primary feature of the common cold. That is called rhinitis. Rhinitis, why? What is a rhino? What is the feature of the animal, a rhino, that makes it remarkable? Is what's on its nose, right? That's why it's called a rhino. Rhino means nose. So rhinitis is inflammation of the nasal mucosa. The common cold seldom causes fever. You may have a mild fever, but never a severe fever. And in addition, it usually does not cause a headache unless there is a secondary infection, for instance, of the sinuses. But usually there is not a strong headache associated with the common cold. It can cause muscle ache, and especially it can cause fatigue. So very rarely does it actually cause a cough due to the viral infection. There may be a secondary cough arises, but it's not actually due to the virus itself. Not usually, anyway. There is variability. And the reason that there's variability in these symptoms from one time you have a cold to another time you have a cold is that, in fact, the common cold can be caused by more than 200 different viruses. And they tend to produce this kind of syndrome. And the common cold is usually of relatively short duration. You usually actually you have a prodromal phase when you feel iffy for a couple of days, then usually about 5 days of actual real illness, maybe 2 or 3 where you really should be in bed, and then about a week recovery, convalescence, but seldom with any sequelae to worry about. So, it used to be believed, by the way, that that the cold was, in fact, having a cold was actually healthy. In the Quran, Muhammad says, they told me I was mad, even though I had had 2 colds that year. It used to be believed that you should have 2 colds a year. And in fact, there's some basis to this. It appears that, in fact, having a cold is a good stress reliever. And probably mostly because it gives you a good excuse, a very good excuse, to get to bed for a couple of days and just relax. And that is the one thing that you should do when you have a cold, not wander around spreading your virus everywhere. So this is what's shown here is a rhinovirus, again, rhino, the nose, and it's acquired from various means. It can be acquired definitely through droplets, as people coughing, sneezing in your vicinity. But rhinovirus actually lasts pretty well on a surface. So it is fomite transmitted as well. And that is usually because that is a very good reason to carry hand sanitizer with you, especially in the cold season, because rhinovirus and other viruses as well can last a long time on a fomite. And you pick it up, and without thinking, you scratch your eye, pick your nose, or suck your finger, whatever, and you can acquire the virus very easily. It does not require a very high dose. It's a very infectious virus. So the reason that they so rapidly extend through the upper respiratory tract is that the temperature in the upper respiratory tract is rather lower than it is in the rest of the body, and that's around about 33 degrees Celsius, as opposed to 38.5 in the rest. This at the bottom here, this is a really, really important feature of these viruses and viruses like them that cause the common cold, that in cold season, you should be very aware of putting your hands anywhere near your face. Your hands can pick up virus all over the place, and it lasts pretty well on your hands, and you then transmit it into your mucous membranes, and the virus will take off. So here's another one. This is adenovirus, and adenoviruses also produce symptoms of a common cold, and the difference, adenovirus more frequently causes a fever. It is a febrile virus. That is, it can cause a fever. And it also causes very painful sore throat and cough. And it may proceed to a viral pneumonia as well, especially in children and in the elderly. And a viral pneumonia is any condition which causes fluid to accumulate in the alveoli of the lungs. We refer to that as consolidation. And obviously, if they're filled with fluid, the alveoli are not going to work properly. And this is one of the viruses that can directly cause a viral pneumonia. Adenovirus also, I've mentioned before, can cause a meningitis. So this is one of the nastier cold viruses to acquire. There was a minor epidemic of adenoviruses earlier in the year. So that is the cold virus. Those are the cold viruses. Remember, no headache, seldom a fever unless it's an adenovirus when you may have a fever. It has a long prodromal phase. You will have 2-3 days in a common cold when you are aware that something is brewing before the symptoms actually break out. This is in direct contrast to influenza. And influenza is not only the confusion between influenza and the common cold arises because influenza also has upper respiratory tract infections. The common cold is by and large an upper respiratory tract infection. Influenza is upper respiratory tract and lower respiratory tract. And in fact, influenza is a very dangerous disease. It's one of the most dangerous diseases in terms of human mortality throughout our history, where the common cold seldom causes problems. Influenza is very, very highly communicable, and it's transmitted by aerosol droplets. You don't tend to acquire it from fomites. And it causes periodic epidemics in the human population for reasons that we'll discuss in a minute. Now, the fact of the matter is that influenza is in fact a zoonotic disease. It is a disease which historically has passed through animal reservoirs into the human population and may pass from the human population into animal reservoirs as well. This is an important feature of its evolution. And as it goes through these passages through animal reservoirs and then human reservoirs, it can acquire changes which make it extremely dangerous, with a very high morbidity and a very high mortality as well. The animal reservoirs that it passes through are pigs and poultry, ducks and chickens as well. So it can, geese as well, but more commonly duck or chickens. So here are the differences in the disease symptoms that distinguish it from the common cold. The first is it's commonly associated with a very high fever. The second thing is that influenza has a short prodromal phase. In fact, people may not even, it has an incubation phase, but the incubation phase seldom produces any prodromal symptoms. And the onset of symptoms is extremely sudden, and it is most frequently the primary symptom initially arises is a headache, a severe headache. And the third thing, sorry, about influenza is it causes systemic symptoms, which are much more severe than in the common cold. Muscle aches, joint aches, extreme fatigue. And then it is not only an upper respiratory tract infection, sharing symptoms with the common cold, it is also a lower respiratory tract infection. And it causes cough, it can cause inflammation of the lungs, and it can directly cause a viral pneumonia. The other thing about it is influenza can be extremely damaging to the mucous membranes of both the upper respiratory tract and the lower respiratory tract. And it predisposes to severe bacterial infections. So that, remember, the distinguishing features, first of all, common cold, upper respiratory tract, long prodromal phase, seldom a fever, seldom severe headache. Influenza, it's a short, very, very short prodromal phase, if at all, sudden onset of symptoms, usually a very severe headache. It's associated with high fever, and it has a lower respiratory tract component to it as well, which may be extremely serious. So the way in which the influenza virus is constructed is a little bit different to most other viruses. So first of all, it's an enveloped virus. Here's a model of it. You can see the envelope around the outside. And then on the interior of this membrane, it has this strange structure. It actually has, its genome is cut up into 8 pieces, 8 different pieces comprised in the whole genome. And each of those pieces is enclosed in its own capsid. It's a very strange organization. On the outside, it has the typical spikes for a virus so that it can attach to the correct cells in the correct host. And these are called H spikes, which stands for hemagglutinin. And hemagglutinin is a binding protein and attaches the cell to the host cells, and also allows it to penetrate into the host cell and inject the 8 capsids. So as we look at influenza as it circulates around the world and as it circulates through animal population, animal reservoirs as well, we have recognized so far, we have recognized 17 different kinds of hemagglutinin. So, and these are numbered, they're numbered H1 to H17. So the other protein associated with the membrane of the virus, of the virion, I should say, is neuraminidase, and that is labeled N. And again, there are 10 types of N, N1 to N10. Then this is so far, because there may be new, we may ultimately recognize more of these. The list just gets longer. So here is the origin of the first of the typing of influenza viruses, because you'll realize with 17 H's and 10 N's, you have a considerable number of combinations of H and N. And so that's why you may hear a virus referred to, for example, as an H1N1 virus. It has H1 and it has N1 in its spikes around, embedded in the membrane. So this is you'll notice here it says influenza A virus. There are actually 3, I'd say 3 species of influenza virus. And these are influenza A. That's by far the most important one. This is the one that we see every year circulating through human populations. And this is one that causes human beings the most problems. There is another one called influenza B. It does arise every year, but it's much, much less common than A. And influenza C, it causes a mild illness, but it is not an epidemic virus. The ones that we are really worried about in influenza are those that arise and spark off epidemics and pandemics in the human population, some of which have had huge, massive mortalities. There are definitely complications associated with the influenza virus, even if you if you're able to recover from it. First of all, secondary bacterial infections, that's one thing. But it's also associated with a condition called Guillain-Barré syndrome. And Guillain-Barré syndrome is a paralytic disease of the nervous system and is probably an autoimmune disease, which can be sparked by various things. But influenza is one of the things which can spark off a Guillain-Barré reaction. The other thing, I'll mention for those of you who have children, you have probably been told this by your pediatrician, whatever. If your child has a fever, which might be due to influenza, do not ever give them aspirin. Seek advice about what to give them, but do not give them aspirin to lower the fever, because it can produce this syndrome called Reye's syndrome, which is potentially very dangerous. So we need to understand why it is that influenza virus makes this regular progression through the whole human population. Because you'd think, okay, these viruses have been around for a very, very long time, and surely we would have built up resistance to the viruses as they circulate every year, and then it should just fall down to a grumbling background. And instead, influenza surges through the human population worldwide every year, and in some years causes pandemics with very high morbidity and very high mortality as well. It varies tremendously from year to year as to what degree of disease influenza viruses actually cause. So the first thing to understand is that as the virus makes its way through the human population, let's say, remembering, for example, that the Southern Hemisphere has its winter while we are having our summer. The virus survives better during winter. The reasons for that are not exactly clear. It may have something to do with the fact that aerosol droplets last longer, or that the cooler temperatures protect the virus when it's airborne or on making its way from host to host, let's say that. As the virus moves through the population, it accumulates mutations in its genome. Those mutations tend to impact the outside proteins, the H and the N proteins, and they produce very small changes in those. But those small changes in H and N proteins are enough that the immune system, which learned about an older virus from last year, let's say, is unable to recognize the new virus when it comes through. So this is not a major change in disease symptoms, not a major change in the degrees of morbidity or mortality so much as they are changes in degrees of infectivity. And there may be additional changes, of course, additional mutations that accumulate. But the most important changes are those that we see in these proteins, and they affect the ability of our immune system to recognize the presence of the virus. That is why we have flu vaccinations every year. What scientists do is they track the virus, especially, for example, in the Southern Hemisphere, in China, where these flu viruses often arise, in the Southern Hemisphere during the winter. They track them and try to spot the new strains as soon as they arise, and they make a decision. There's a committee meeting where they look at all of the data about the different flu viruses they can see, and they make a decision about at least 3 strains which they think have the potential to become pandemic. So they may spot them locally. They may spot them in Sydney, Australia, or in Cape Town in South Africa, or in Hong Kong or somewhere. They see a new strain and have to assess, does this have the potential to cause a pandemic? If it does, that is one that they will select for the vaccine, for the new vaccine. The vaccine is very rapidly produced. It's not a live vaccine. The vaccines you get have no live virus in them, but they're usually against at least 3 strains of flu. Some years they get it right. This year, apparently, they have included the most important of all of them, which has high potential for pandemic spread. So this year's vaccine is a very effective vaccine. Other years, it's a hit or miss. So some years it's not as effective. So this is what we deal with year to year, is the slow change, and that is referred to as antigenic drift. But there's a much more dangerous situation that can arise. Remember that I said, that there are 2 very important reservoirs of the disease. The first, probably the most important, are in birds. And there are many bird flus which circulate through bird populations back and forth. But of course, there's one set of birds which we have intimate contact with. And these are the poultry. This is chickens, ducks, geese. These sorts of birds are farmed and are dealt with on an intimate basis. And believe it or not, birds sneeze, and they release droplets into the air. There is another reservoir, and that is pigs. Pigs are particularly important because they act as a mixing vessel. They can acquire virus both from birds and from human beings. So we have a situation now. Let's just look at the birds. We have a situation here where they have their own influenzas that circulate through the bird populations, acquiring mutational changes, etc., from year to year. And some years they're much more dangerous to bird populations than in other years. And there is a possibility, first of all, that we can acquire bird influenzas directly. So this now is an entire possibility that we will acquire an influenza virus which is radically different to anything. These ones here, our immune system can pretty quickly adapt to a virus that has drifted. But these here, if we acquire from an avian population, we have absolutely no resistance. We have no basis for resistance because our immune systems just haven't seen that kind of virus before. It's an entirely new species of virus to our immune system. The second thing is that the virus can pass to pigs, and it can be further adapted in the pigs, and then transmitted to humans. It's the same story. What is happening now is that we are acquiring viruses with entirely new genomes that we have never seen before, passing from the reservoir directly. But there's an even more dangerous situation. And that is that the pig acts as a mixing vessel, and it acquires virus from birds, and it acquires virus from human beings. And these are reassorted. Remember, there are 8 elements to the genome, each enclosed in their own capsid. These viruses, human, bird, can infect a pig, and those capsids can be mixed up in the construction of the viral particles during the cell cycle. And now we've got a major antigenic change, a major antigenic change. And this is the source of really virulent pandemics of influenza. And we have seen numerous of them during human history. And they've usually arisen through this route here, either directly or through genetic reassortment. And that is most commonly in pigs. So here, if we look at some of the major influenza epidemics, this is what we see. Look here, this is an H1N1 influenza virus, and it was acquired directly from birds. No, there was no pig reassortment involved. All this is a bird flu, which had adapted so much in the bird population that it could actually infect humans, and humans could then infect other humans. So this is an important feature. It's one thing if somebody acquires a bird flu virus and they may get very sick and die, but be unable, but the virus cannot be passed from human to human, then there is no epidemic. It stops right there. But if human beings are infected with a bird flu or a pig flu and can pass it to other humans, then you have the potential for epidemic and for pandemic. This one is a very famous one. H1N1, the bird flu, infected the human population in the mid-1900s and produced a pandemic which killed millions and millions and millions of people worldwide. And this was the famous, it was called the Spanish flu. It had nothing to do with Spain, nothing at all. It probably arose in China originally. Sorry, it probably actually arose in America initially and was disseminated through, largely through the movement of men going to fight in the World War and then returning home to the United States or to other countries, and spread worldwide as one of the greatest pandemics that the human population has ever seen. So this is the so-called Spanish influenza. 1918 was the major mortality, but it had been around for some years before then. So in this case, there's no reassortment. This is a bird flu, plain and simple. But it's a bird flu that had enough mutations that it could allow human-to-human transmission. Here is another one. This is H2N2 virus, and it's actually a reassortment of an avian virus, a bird flu, and a human virus, an H1N1 human virus, in pigs. And here it is here, and it caused the Asian flu, another major epidemic here. This was in the late 1950s. So there was another one called the Hong Kong flu, only named the Hong Kong flu because that was where it was first recognized, but it had actually arisen in China. This was H3N2, and this was an avian virus and a human virus, which again reassorted in pigs, and there it is there. This one here is a little bit different. This is a relatively unusual flu virus. It was actually a pig virus, whose genome became mixed with bird and with human. Pigs can acquire and pass influenza viruses of their own, but they don't commonly reassort with it, the reassortment in pigs is usually avian and human. But in this case, the new virus acquired parts of the genome of each of these. And it caused a grumbling series of minor epidemics. Okay, so the take home is that this antigenic shift is a major change in the genome of the influenza virus, usually due to reassortment or because a new virus has been acquired directly from an animal reservoir. And influenza remains one of the great causes of mortality in the human population. I should just tell you, the stories from the Spanish flu are quite horrific, but they point to the fact that influenzas tend to have a very short prodromal phase. So many stories from the Spanish flu of people waking up feeling completely normal, going out about their day and collapsing on the street, and having to be rushed home and living only a matter of hours or days before they die. One of the reasons for that is that these virulent flu viruses tend to cause a very, very powerful immune response. And very often people are actually killed by what is called a cytokine storm, as elements of the immune system chatter at one another and positively reinforce their activity to such an extent that it causes a lethal condition. Okay, so we have, of course, got to talk about the other respiratory virus that we've had to live through the great pandemic. The pandemic we have just lived through, by the way, rivals the Spanish flu in mortalities. And this is testament to the power of viruses, of new viruses, which enter the human population for which humans have no resistance. Now, the coronavirus are a very well-known viral family, and they appear to mostly arise from an animal reservoir, so their origin is zoonotic. And that animal reservoir, the ultimate animal reservoir for these viruses, seems to be in bats. Why exactly that is true is not known, but bats are very, very tolerant of viral infections. And they harbor and keep latent many different viruses. Exactly why we don't necessarily understand. But this, the current epidemic is caused by SARS-CoV-2. That stands for severe acute respiratory syndrome. It sounds like overkill, but there was actually a syndrome recognized called acute respiratory syndrome. And this is the virus that gave this name was severe acute respiratory syndrome. And the original virus was SARS-CoV-1, and I'll tell you about it in a minute. The current virus is SARS-CoV-2, but it closely resembles SARS-CoV-1. And this is probably familiar to you. It's an enveloped virus. It is an single-stranded, positive-stranded RNA virus. And here's the envelope on the outside with all of the viral proteins embedded. Those viral proteins are what bind it to its receptor cells and determine the host range and determine which cells in the host are going to be infected. Now, the coronavirus is a big family of viruses. And there are, in fact, 3 coronaviruses which have been recognized for a long time as cause of a common cold. They cause a mild illness of no great import. But new coronaviruses which enter the human population seem to cause extremely serious disease. It is probable that the 3 coronaviruses, which cause mild illness, also once upon a time caused tremendous morbidity and mortality in the human population. But then we adapted to them, they adapted to us, and they now just form the background common cold virus. The new coronaviruses are associated often with very severe illness. And the first one was SARS-CoV-1. Then there are some other strange ones. This one here, MERS, is Middle East Respiratory Syndrome. And it is acquired, the human beings acquire it from camels. But it is probable that these animal vectors here, animal reservoirs rather, acquired originally from bats. So the SARS-CoV-2, it is not yet clear how we acquired it, how it entered the human population. That is a matter of speculation, but it is believed to have been from animals that were being sold in an animal market in China. So these two, SARS-CoV-2 and MERS, are extremely virulent viral diseases with a very short prodromal phase. This is important. I'm sorry, with a longer prodromal phase, but very sudden onset of symptoms. And the symptoms, when they appear, are extremely violent. People actually tend to become very ill very, very quickly, and they have a very high morbidity rate, SARS-CoV-1 and SARS-CoV-2. I mean, SARS-CoV-1 and Middle East Respiratory Syndrome, MERS-CoV. So these two have the epidemic possibilities are relatively limited because the disease onset is so recognizable and it is so rapidly fatal that it limits the ability of somebody who's infected to spread it to other people. SARS-CoV-2 is a bit different. It has a long prodromal phase in which people can actually infect many others. And it certainly has the potential for being fatal. I mean, we know that we've lost more than 1 million people in the United States alone died because of SARS-CoV-2. But many people could actually carry on functioning, and that's important because those people who felt ill, were symptomatic, were sneezing, coughing, could still circulate in the population and cause the spread of the virus, leading to the pandemic that we've just seen. So it is worthwhile just looking at SARS-CoV-1 and seeing some of the differences, not only in the disease process, but in the epidemic process. So the SARS-CoV-1 is spread only by fairly intimate social contact, by being close enough. For example, many of the people who ended up being infected with SARS-CoV-1 were healthcare workers. This was an unfortunate and very tragic effect of this disease when it entered the human population. It's a very, very virulent virus. It has a high mortality, 14 to 15% overall, and 55% or greater in people over the age of 60. This is unusually high for a viral disease. You think about it this way. It's of no advantage to the virus to kill its host so quickly, because then the host does not have a chance to spread the virus when the new viral particles are produced. This is characteristic of disease of new viral species entering the human population. So the disease could be spread by contact, but it could also be spread through fomite contact. So if people were sneezing around you with SARS-CoV-1, you could sure acquire it, but it also could be picked up from surfaces. This has limited validity for SARS-CoV-2. There's very little evidence that it is spread through fomites. There, in the 2002-2003 epidemic of SARS-CoV-1, 8,000 people were infected, 774 died, and most of them were healthcare workers, which is a very sad aspect of this. So one of the things about SARS-CoV-1, though, the disease onset was very sudden, but the disease symptoms were so distinctive that it was very quickly realized that this was a disease where some people had to be put into isolation, into absolute isolation immediately. And the news about the disease spread so rapidly that people were terrified and immediately reported as soon as they felt ill. So here's the major difference. In SARS-CoV-1, is the short prodromal phase with a very high morbidity, and that allowed rapid identification and isolation. That's not true for SARS-CoV-2. It has a long prodromal phase when people can wander around spreading the disease. It has, even when people are ill, when they're producing viral particles, and they're symptomatic, many people could still carry on functioning, and they could spread it very, very widely. SARS-CoV-1 was a self-limiting epidemic. And here, I'm not going to do this in any great detail, but it arose in much the same area as SARS-CoV-2, but in China. And it very rapidly was transported to Hong Kong, which was the major center of spread, because Hong Kong was the financial center, and it's a center of international travel. So people who acquired the disease then traveled to other countries. Most of the cases of SARS-CoV-1 that were spread internationally, the people arrived with the disease and were so ill that they were immediately isolated. So its spread globally was very, very limited. That was to the human being, to our advantage, of course. It had such a high morbidity and mortality that it had huge impact for people, for that population who were infected. But ultimately, only 8,000 people were infected, and the epidemic soon terminated. In the meantime, it had spread all over. There were some instances where in foreign countries where people spread it from person to person, but they were very, very limited, and it was usually to healthcare workers. So SARS-CoV-2 is an enveloped, again, it's very similar to COVID-1, but it's an enveloped positive stranded RNA virus, single strand RNA, and the capsid is actually a helical capsid. You can see it there, coiled up inside the envelope. Here are the envelope spike proteins. And those spike proteins are tremendously important because they their structure forms the basis of our vaccines against SARS-CoV-2. So this is the way it gets into cells. Those spike proteins bind actually to a particular protein here. This is a receptor, it's an angiotensin receptor. It's a hormone receptor, never mind about the details. But it is a receptor embedded in the cell membrane of many cells of the body. And the virus binds to it. And what happens is it induces the cell to swallow the virus in a vesicle. So here you can see the vesicle budding into the cytoplasm. So that is the way it gets in. Here's the viral particle. the virion, and here it is in the membrane derived from this cell membrane, forming this vesicle. But what happens now is the envelope unites with this membrane here and opens up to spit the capsid, this helical capsid, into the cytoplasm. The capsid proteins are digested off by the host, because the host cell recognizes foreign protein and digests it with proteases, but liberates, of course, the genome. Now, the genome is a positive strand, mRNA, and it's immediately translated by the host ribosomes. So the first thing that happens when it's translated is that it produces a special polymerase, a special RNA-dependent RNA polymerase that reads off many copies of the viral genome to produce many copies of the viral genome in the cytoplasm. The other part of the genome, of the RNA genome, is read off just as if it was messenger RNA, and it produces the viral proteins, but it does so in association with the endoplasmic reticulum and with the Golgi apparatus. And in doing so, it produces all the viral proteins, which will enclose the genome, and it produces all of the elements that will be embedded into the viral membrane, into the envelope of the virus. And that envelope is now derived from the membrane of the Golgi and of the endoplasmic reticulum. So here is the membrane here. This originated in the endoplasmic reticulum and the Golgi, but it has viral protein embedded in it. Here is the genome being packaged into this membrane, and that then forms the new virion. And this vesicle that it's in here moves the virus to the cell membrane again, where it is liberated. This is a process of exocytosis. It comes in by endocytosis and it leaves by exocytosis. So you'll realize how critically important these proteins are here embedded in the envelope, because they are what govern the virion's ability to enter its host cell and be replicated through this complex process. That is why the vaccines that we use are directed against those proteins. They are, first of all, they're essential to the virus's function. If you can gum them up somehow and stop them working, then the virus can't get into cells to be reproduced. But second of all, they are visible to the immune system. They are what's on the outside and can be seen by the immune system. Now, the old-fashioned way of producing vaccines would be, for example, to produce a solution of these proteins, perhaps, and inject it into an animal and get its immune system to produce antibodies, and then harvest those antibodies to be used in a vaccine or whatever. Or else, so what we could do then is we could actually take these spike proteins, render them non-functional, and inject them into a human. It's just another animal. Inject them into a human, and our immune system would produce antibodies to it. Unfortunately, that is not actually nearly as simple as it sounds, and that kind of vaccine does not always produce a very strong reaction. There was another system of producing the vaccine, which was totally innovative, but had behind it at least 10 or 15 years of work. And this is to take a completely different approach. And these are the most influential vaccines that we have seen, and these are the mRNA vaccines against SARS-CoV-2. First of all, remember what messenger RNA does. Messenger RNA, when it enters the cytoplasm of a cell, by whatever means, is recognized by the ribosomes, which translate it to produce proteins. So if we can somehow get mRNA, which can be translated to those spike proteins, into a cell and get them to liberate those spike proteins, then the immune system will see the spike proteins in circulation and act against them. And in doing so, the immune system forms a memory of those spike proteins, so that if they ever see them again, they will react against them. The question is, how do we get those instructions, the mRNA instructions for the spike proteins, into the host cells? And the answer is, this was the whole technology of this vaccine system. They built vesicles. They built special vesicles, artificial vesicles, with proteins in the outside, which would recognize the host cells, bind to them, and unite with the cell membranes. So this is the important technology. The important technology was the creation of those vesicles, because once you produce those vesicles, you can put whatever mRNA you like inside. Recipes for virtually any protein. But this has first been used for SARS-CoV-2. Here is the vesicle that's going to be injected. It's entirely artificial, and a solution of these vesicles is made in such a way that they enclose messenger RNA, which contains the code for spike proteins. These vesicles are injected into you when you get your vaccine. They unite with your cells, usually right in the area of the injection site and somewhat in the lymph nodes nearby. And they induce cells to produce spike proteins. And those spike proteins go into circulation, where they are recognized by the immune system. And they produce antibodies to them. They disable those spike proteins, but they also form memory. And they form memory of the structure of the spike proteins if they ever see them again. If, for instance, you acquire the virus through droplets or whatever, your immune system recognizes those spike proteins and gums them up with antibodies, so that the virus cannot attach to its host cells. The huge advantage of this system, and the reason it was developed, was this is the technology, this is the difficult technology, creating that vesicle, not getting the messenger RNA, not putting the messenger RNA inside. It was creating this vesicle. Once that is done, you can use that same vesicle for many different kinds of messenger RNA, for many different diseases. That is the hope of this technology. So I also just want to mention quickly another virus, a respiratory virus that you will have heard of. And this is respiratory syncytial virus, RSV. And the RSV in adults, usually the infection is very limited. It's usually an upper respiratory tract infection and may often appear like a common cold. But in children, it can be extremely dangerous. And children and infants, it can be extremely dangerous because it moves to the lower respiratory tract and it causes severe bronchitis, also bronchiolitis. Bronchitis is inflammation of the major bronchi. Bronchiolitis is inflammation of the smaller airways in the lungs. And ultimately, it can cause a very severe pneumonia, and it has a high mortality, can have a high mortality in children. Up till now, there have been no effective vaccines. But there are 3 vaccines which are now coming online, which appear to be effective. So if you have children and you're concerned about respiratory syncytial virus, then keep your ears open, talk to your providers, because these effective vaccines will soon become available if they're not available to you already. Okay, so let's now turn our thoughts to the herpes viruses. I know that you obviously have heard about herpes viruses before, but they are really important human viruses. But in general, they do not cause life-threatening illnesses except in special circumstances. So the herpes viruses are a very, very diverse group of viruses, very large and diverse group of viruses. And one of the characteristics is that they very frequently, after an initial acute infection, will form a secondary latent infection, which may or may not revive as an acute infection. So the first group, ones that you have surely heard of, and these are the simplex viruses, the herpes simplex viruses. And the herpes simplex 1 is the virus that causes cold sores on the lips and face. And it behaves in typical herpes fashion. It is acquired it can be acquired through touch, not through aerosol, anything like that. But it requires a somewhat more intimate contact. And not even necessarily kissing. Kissing certainly will spread it. But if you have touched somebody who has the virus and then touch your own face, that is sufficient to cause the initial infection. And of course, in children, that is very frequently the case, that children interact with one another, hugging, kissing, playing, whatever. And that is usually where we acquire our initial dose of herpes simplex. And once acquired, then it causes an ulcer, a painful blister that we refer to in herpes simplex 1 as a cold sore that heals over and disappears. But it has actually gone into a latent phase in the nerve endings of the facial nerves. And it can revive at some point in the future, or else remain entirely dormant for the rest of your life. But it is still there as a prophage in the nerve cells of your face. The reason why it revives again and causes a second acute infection is unclear. It's often attributed to stress, also to the cold, which may actually be a factor. Any damage to the tissues might spark it. Herpes simplex 2 is a very, very closely related virus. It's a strain of the same virus, which causes genital herpes. And I'll discuss that in a bit more detail, but in actual fact, they can be interchanged. You can have cold sores produced by herpes simplex 2. You can have genital herpes produced by herpes simplex 1. The other important herpes virus in human populations are the varicella viruses. And this is the virus which causes chickenpox. And varicella zoster is the name of the virus that causes chickenpox. Now, chickenpox is actually acquired through aerosol as well, like so many other viruses, and is most important in children as a childhood disease, and also in later on in life, because again, it can become latent and make its reappearance later in life. as the disease of shingles. The others, I only want to mention this one here, Epstein-Barr virus, and that is called Epstein-Barr virus. Actually, it's a family of viruses, the Epstein-Barr viruses. There are various strains of the virus that cause different things. But the one that you may know of is mononucleosis, which is referred to as kissing disease in high school, but in actual fact, easily acquired, even if you didn't kiss somebody. But the last one is Kaposi sarcoma virus. Now, this is a very interesting one because Kaposi sarcoma is a disease which is only occurs usually in very elderly men, Jewish men of Ashkenazi descent. Very limited population. And it's almost unknown outside of that population. And it never kills them. It causes a tumor, it causes a cancerous condition, but it never kills people. It's so slowly progressing. But this is the disease, Kaposi sarcoma. This is one of the diseases which mark the start of the AIDS epidemic. This is one of the infections which was a hallmark of the collapse of the immune system in young men of all ethnic backgrounds who were suffering from AIDS. And it's a testament to the fact that many of us may have these viruses that we've acquired somewhere along the line, which never make an appearance because our immune systems recognize them so quickly that they are never able to establish infection, or if they manage to establish infection, they can never reoccur. But in these young men with collapsed immune systems, this was one of the diseases which the CDC spotted early on and first identified as a syndrome, as part of the AIDS syndrome. Okay, so yeah, these are cold sores, HSV1, and it's very typical. This is a little bit confusing, this diagram. This is the actual viral, the virion here. These are the virions there. And these are DNA viruses. The herpes viruses are all DNA viruses. And this is typical cold sore that you see, and you probably many of you have experienced. So the genital herpes it differs between men and women. In women, they may not actually be aware of the fact there may be some irritation or some suppuration on the vagina. They may not be aware that they have an active lesion due to herpes simplex 2. But men notice very, very quickly because the lesions, the acute lesions of the initial infection appear on the penis and also cause urethritis, pain on urination. And nothing sends a guy to the doctor quicker than painful urination. And the blisters, unlike the cold sore, where there just tends to be one or whatever, the blisters can be widespread and are intensely painful. So in men, they tend to seek treatment very early. In women, often the infection goes almost unnoticed, and this can have dangers. You're only infectious when you actually have active lesions, because these lesions are produced as part of the reproductive cycle of the virus. This is how the virus is disseminated, is through these lesions. If you do not have active lesions, then you're not infectious. In women, they may have lesions and not realize it and actually be infectious. Now, a couple of things to be said. First of all, you can acquire HSV1, the cold sore virus, through oral sex. And you can also acquire the cold sore, what looked like cold sores, as a result, especially of oral sex, on the face from HSV2. So the two are kind of interchangeable, but then the net effect is pretty much the same. The thing is that, of course, the occurrence of lesions is going to interfere with somebody's sex life, but also in women, the problem is that they can transmit the virus to a child who is being born through vaginal delivery. If they have active lesions, they can transmit it, and it causes. First of all, the one thing it can do is cause herpes keratitis. This can occur in anybody, by the way. You can, if you get the virus established in the eyes, it can cause this keratitis and inflammation and scarring of the cornea. But this is a particular danger in newborns. And the virus can actually disseminate through the body of the newborn and cause all sorts of neurological problems as well. So it's very important that obstetricians take a very careful history to see if, and may even test a mother-to-be to see if she has antibodies to HSV2 in order to take precautions. And precautions can be taken. There are in fact some antivirals, which are not a cure, but will shorten an episode and also can prevent, be taken prophylactically. So mother-to-be who has had previous infection, she can be put onto one of these antivirals to see her through the birth, to ensure that she doesn't transmit it to her baby. Okay, so this is varicella zoster, and this is chickenpox. This is typical chickenpox rash, and these are pox. These are pox. They're the little pustules, and it's very, very, very highly transmissible, highly communicable disease, and very rapidly spread. Will very rapidly spread through a population of children, and if it again transmitted by direct contact, skin on skin, but also by respiratory droplets, which is a little bit unusual for the herpes viruses, which usually require intimate contact of some sort. But in children, it can actually turn into a very serious illness and a life-threatening one, but it does not have a high mortality in otherwise healthy children. But it can definitely have sequelae, and it should be regarded as a serious illness in children. In adults who have had chickenpox, the disease, again, behaves just like any other herpes virus. It can become latent and reside for years, especially in the nerve endings of the vagal nerve. And this leads to it producing this disease of shingles. And shingles can result in, I'll show you in a second here. It produces this band of lesions here, extremely, they don't look like the chicken pox. Instead, they are more like cold sore kind of blisters, intensely painful. It can also occur in the face. It can occur in the facial nerves as well. And there is a certain incidence afterwards of chronic pain that can arise from a dose of shingles. And shingles can make its appearance more than once. But there is a good vaccine called Zostavax, which you can have, which will prevent the reoccurrence of the virus as shingles. Okay, so let's talk about a couple of the other diseases. These are diseases, the herpes virus, simplex virus, varicella. These ones are diseases of the skin, but often acquired again through respiratory droplets. So now we're finished with the herpes viruses, okay? These are other viral groups. And this is a group of viruses called the paramyxoviruses, which cause several childhood diseases. The most important one of all is measles. And measles is also called rubeola, because the rash that measles is named for, the actual measles, causes reddening of the skin. And there can be so many of them, they become confluent and form this rosy red rash. Measles is acquired primarily through respiratory droplets, and it is one of the most highly communicable disease of all human diseases. It is also a very dangerous disease in children. It has many complications that can become associated with it, and it has a relatively high mortality in children as well. It's a very serious illness. And in the developing world, it's a major source of infant mortality, and infant and childhood mortality. A very good vaccine exists, and this is the MMR vaccine, measles, mumps, and rubella vaccine, each of which we'll discuss. Now, quite apart from the fact that it's inherently a dangerous disease, measles can also produce, I have mentioned this before, it can go into a state where it remains active in the body at a very, very low level, and years later, if not decades later, can cause this terrible neurological disease, which has about 100% mortality, called subacute sclerosing panencephalitis. And that's also called Paget's disease. And it's an inflammation, an infection and inflammation of neural tissue of the brain. A very nasty disease. So this vaccine, the MMR vaccine, is a very, very important vaccine. It resulted in almost elimination of measles in childhood populations in developed countries, especially here in the United States. But unfortunately, as you probably know, some doctor in the United Kingdom produced an entirely fake medical paper linking this vaccine to autism. And there is no association whatsoever. Millions upon millions upon millions of research dollars have been spent examining this question. And all of them, no matter who did them, all studies have come out very strongly saying there is no association between this vaccine and autism or other syndromes that they have ascribed to the vaccination. But unfortunately, as a result of that, the activity of anti-vaccination groups, measles is now making its reappearance in epidemic form in areas of the country where these anti-vax groups are particularly active. And we will see in those areas, we will see an increase in Paget's disease as well, undoubtedly. So mumps is another, it's actually a somewhat allied virus, but it causes, in general, mumps causes a relatively mild illness, which is characterized by these enormously swollen parotid glands salivary glands underneath the jaw, and it produces this kind of bull neck look to people. Again, spread by respiratory droplets, but also by fomite as well. And it's in general in children. There are always exceptions, but in general, it's in children, it's a self-limiting disease. And but it is included in the MMR vaccination, because if you miss out on having mumps as a child, or if you have had mumps as a very young child and then much later in life, you are exposed again. In adults, it can produce some very serious illness. And in the best known effect is in men. It produces orchitis, massive swelling of the testicles, which doesn't always, but it can result in infertility. But it can spread further than that. It can also cause pancreatitis and produce, as a result of that pancreatitis, produce diabetes. But it can also cause encephalitis in adults, which is extremely serious and can actually be fatal. But again, this is a very, very good vaccine, the MMR vaccine. And it has virtually eliminated the occurrence of mumps in the population. Now, the last one is rubella. This is a different virus, and rubella is called German measles. In children, it causes a short-lived acute infection. It's not particularly infectious. It also spread by respiratory droplets or by contact. And it in children, causes a mild fever and a kind of a generalized rash, but it's a short-lived illness. And it's in children, it's not. So you have to ask, why then, if it's not that serious in children, why are we giving the vaccine? Here is a problem. And that is that adults who have not been vaccinated can acquire rubella. They can also have a recurrence of an old rubella infection, but still mild. And this is particularly dangerous in women who are pregnant, because it can cause all sorts of problems in the fetus. It can cross the placenta, and it ends up causing eye, ear, heart, even deformities of the extremities. It can have very, very, very serious consequences for a fetus. And again, the MMR vaccine has virtually eliminated this in the population. But where the vaccine is not being given anymore, you can expect these things to reoccur. Okay, so here's another one that's of considerable importance. And this might be of importance especially to college students and to young people who are first entering into sexual relationships, and is maybe exploring around or whatever. It's something to be very, very much aware of. And this is human papillomavirus. Now, the human papillomavirus, there are many strains, different strains of the virus, but the one that we all know is the one that causes common warts. And these occur on the limbs, on the fingers, etc., and usually self-limiting. And they are very easily spread, especially amongst children, amongst school children, because they can also be picked up from fomites, from school desks and everything. But it's usually a feature of childhood, and our immune systems learn to recognize the presence of the papillomavirus, and they eliminate the warts, and in turn they become the body becomes immune to that particular strain Remember, this is a particular strain that causes the warts on the body. It is an entirely different strain that causes genital warts. And genital warts, again, are very easily acquired, especially through sexual activity. And the virus is extremely common, even if it doesn't manifest itself as genital warts. But genital warts often produce enormous warts on the genitalia, so they are relatively easily spotted. That is the one manifestation. Those are not of particular importance. It's also become self-limited, although they may take a long time before they disappear. But they can be burned off, they can be treated. So that's the one manifestation of genital, but there is a second manifestation of genital warts, of the genital papillomavirus. And this is this is an oncogenic virus. And while you may eliminate the warts themselves, which are referred to as condylomata, the virus itself can be maintained in tissues of the genitalia, of both the penis, but especially in women, in the cervix, and result in tumors. Genital tumors of the penis are very unusual. They're far rarer than changes to the structure of the cervix. And the changes to the cervix result in cervical cancer, can result in cervical cancer. So this is another disease for which there is now a very, very good vaccine, and this is called Gardasil. Works extremely well, and where all the studies have shown that where Gardasil or now there are others as well, vaccines against human papillomavirus are being used, the incidence of cervical cancers plummets. So here's an Australian study. There's a normal background level of production of cervical cancers. There's a sort of a background level. Here is where they introduced the first vaccines against human papillomavirus, and just follow the line. This is the rates of cervical cancer, just plummets. It's really a very, very impressive success in public health. Okay, so we're going to finish off just by quickly discussing human immunodeficiency virus, HIV. HIV is the causative agent of the syndrome called acquired immunodeficiency syndrome, AIDS. And that describes it absolutely. AIDS is an immunodeficiency disease, but it is not a spontaneous disease, that can occur, but it is due to an infectious agent. And the HIV is a positive stranded RNA virus, but it is different to other positive stranded RNA viruses in that it is not read off as if it was messenger RNA by the host cell. Instead, the virus itself contains a polymerase. So here's a diagram of the virus itself. It's an envelope virus. It has receptors on the outside, and you'll notice here it has two receptors that are particularly important. The first is to CD4. And CD4 is a marker of certain immune cells, also of some neural cells, and there are a couple of other cells in the body that have the CD4 protein. There is another receptor, which is for us, it's to a cytokine receptor. So it's particularly important receptor for the immune cells. So those two, this is CCR5, that's its name, the cytokine receptor. and the CD4 receptor. Both have to be bound by the spike proteins of HIV in order for the virion to attach and then inject its capsid. Here's the capsid. Here's a conical capsid. Contains two single strands of RNA, and it also contains this enzyme here, which is reverse transcriptase. And reverse transcriptase is a polymerase, but is a very peculiar polymerase, because what it does is it reads RNA and it produces DNA. And I pointed out in class before, this is the inversion of the central dogma. That is why it's called reverse transcriptase. It's because it reads RNA, but it produces DNA instead of reading DNA and producing RNA the way it usually happens inside the cell. So this is the dreadful virus itself attaching here to a CD4 cell. Now it says here T cell, but not all T cells are in fact CD4 cells. The most important host cell for humans is T helper cell. And a T helper cell interacts with B cells, which actually produce antibody. They interact with B cells to enable them to produce abundant antibody of high potency. So if the CD4 cells die off because of HIV infection, then the ability of B cells to produce antibodies is severely limited. And ultimately, once all of the CD4 cells are eliminated, the immune system collapses. That is the pathogenesis of the HIV virus. Okay, so here we have a look. Here's the virion. Here's the capsid, the conical capsid, two strands of RNA, messenger of positive stranded RNA with two molecules of reverse transcriptase. So it's the usual set of entry steps that should be familiar to you, because the proteins embedded here unite with the cell membrane of the correct cell, CD4, CCR5 bearing cells, and inject the capsid. The capsid is digested off by host responses and liberates the viral RNA. The viral RNA produces a double-stranded DNA copy of the viral genome, and that integrates with the host DNA in the nucleus. Now it goes silent, and it will remain silent until this T cell is actually activated, until the T cell enters into an immune response. And once the cell is activated and its own DNA is being read off, only then is viral RNA produced. And two kinds of viral RNA are produced, as we heard before. The first is the genome of the virus. The second are all of the viral proteins. And they are then packaged and they bud out. They cause viral proteins to be embedded in areas of the host cell membrane. And the virion then buds out so that it is enveloped correctly, and it contains, again, the capsid with the two strands. So I want to just point out to you that each of these steps, attachment, entry, reverse transcription, integration, the production of viral proteins, and budding out, each of these steps provide a possible way of intervening. If we can interfere with any of those steps, we can interfere with the production, with the whole production of the viral particles. And all of these have been tried. A very early initial attempt was made to actually flood people's bloodstream with substances, even antibodies and other substances, which would gum up the spike proteins and stop them attaching. Nothing really worked. The most important next step was to interfere with this process, the reverse transcription of the viral RNA. And that was the use of AZT, azidothymidine. Instead of including thymidine here, it would include azidothymidine, and that would stop the process of reverse transcription. And that works up to a point, but there's a problem with HIV. This polymerase here has no proofreading ability, unlike normal DNA production in the cell. This has no proofreading ability, and the virus mutates very, very quickly and very efficiently to evade drug, especially to evade anything that would interfere with its reproduction. In other words, drugs, it tends to evolve away from the drug. The real miracle in treatment of HIV comes all the way down here. When the viral proteins are produced, they are produced as a single long strand of polypeptide, which is then cut up into pieces by a set of enzymes called the proteases. And these, the proteases actually are very efficient cutting this strand into the correct sizes. So its presence and its activity is essential, because if this strand is not cut up properly, the viral particles cannot assemble. The viral proteins can't assemble, the viral particle can't assemble. And this is our most important avenue of treatment, is to use protease. Protease is an enzyme that cuts polypeptide protein into pieces. We use a protease inhibitor, and that protease inhibitor stops the activity of the proteases and stops the production of viral particles. You still have a problem in dealing with resistance developing, but what is done is that there are a variety of protease inhibitors, and the list is growing. And so they can roll over the protease inhibitors, keep people on a set of inhibitors for a certain length of time, then change the formulation to prevent resistance arising in the viral population. So the impact of a viral infection in a human being follows some pretty well-defined steps. And some of them may surprise you. First of all, here's an initial infection with the virus. The initial infection of virus actually proceeds quite rapidly. And the reason seems to be that it does excite an immune response. So the initial infection of cells in the body is very rapid. If you were to follow the number of cells that were infected, the amount of HIV increases markedly. Here is a peak within about a week or two, usually. There is an acute illness. It's an acute illness which produces fever, very often mistaken for influenza. People can get extremely ill at this point. If we were to look at the number of T cells in the immune system, you see that the number of T cells plummets in this 2-week period. So this is a period of acute illness, but that resolves itself. It will spontaneously resolve itself, because the virus now it retreats. It retreats into those T cells which can continue to survive with the virus present in their genome. If we're now to follow the amount of HIV, we find the virus itself in circulation. The amount plummets, but it doesn't go to zero. if we were to look at the number of T cells, we begin to see now a slow decline in the number of CD4 positive cells as the virus slowly increases. And this may be over a period of many years that this process takes place. In other people, it takes place within a period of months, but in some people, it's a matter of years before they begin to suffer real symptoms from the decline of their CD4 cells. When the normal count, the normal CD4 cell count is between 1,000 and 1,200 per microliter. And the amount declines slowly, and the people may start to suffer symptoms around about here. And they may start to notice that it takes them longer to recover from ordinary viral things like cold or whatever. They may start to see some strange skin things arising. They may start here to have night sweats. It's almost like a prodromal phase of milder symptoms. But when they reach this point of less than 200 T cells per microliter, this is the definition. By definition, they now have AIDS. And as the T cell count plummets below 200, they begin to suffer from the onset of infectious diseases, which under normal circumstances, their immune systems would prevent them from having entirely. They begin to pick up strange parasites. They begin to show signs of Kaposi's sarcoma, of weird pneumonias, of strange neurological symptoms from disease-causing organisms, for example, which normally just cause a mild illness. All of these sorts of these diseases that are normally a healthy body would take in stride using the immune system. Now those diseases begin to look fatal. And indeed, most people who die of AIDS die not as a result of the virus itself, but as a result of all of these secondary infections, secondary conditions, because they lose their ability also to fight tumors and things like that as well. The collapse of an immune system is catastrophic. At the same time, the HIV load begins to increase prodigiously as all of these T cells die and release their virus. Now, the other important thing to realize is that people are infectious right from early on in the infection, all the way through to death. They are infectious, and this is how the disease was spread. It is usually spread, it is usually spread by intimate contact, not necessarily person to person, but in any contact which would spread body fluid from one person to another. So obviously, use of unsterile syringes, drug users would often exchange needles, exchange syringes, and drug users indeed were one of the early high-risk groups for HIV. In pregnancy, a mother who is HIV positive can pass the disease to a child, even if it was not born with HIV. The child can acquire HIV through breastfeeding. Blood transfusion was another source of infection, especially early on. Nowadays, of course, the blood is all tested for the presence of virus. Organ transplants also spread it, but the most important vehicle of all was unprotected sex. Sexual transmission was, and still is, a very, very important factor. Now, one thing to be said about this. Here in the Western world, in the United States, Europe, people are used to thinking of HIV as being a gay disease. Well, it certainly made its appearance in the gay communities in Europe and the United States, first of all, but is by no means a gay disease. In all of Africa and much of Asia, which have huge populations that are infected with HIV, the virus is spread through heterosexual sex, not through homosexual sex, primarily. So it is the fact it is spread sexually. It's just the circumstances in which it enters a population initially. In the Western world, it happened to enter into the gay population. In Africa, it spread through the heterosexual population. Now we test for, in the old days, all that you could do was test for the presence of antibody to the presence of the virus. And that was the meaning of saying somebody was HIV positive, was that they had evidence that they were producing antibody to the virus. But nowadays, much more important we test for the presence of viral RNA. We can test not only for its presence, we can test for the amount that is in circulation to get an idea of how much virus there is actually present. The treatments I've already mentioned. The first treatment was AZT, interferes with reverse transcriptase. It's not a good treatment in the long term because resistance to it develops so quickly. But it's a very good prophylactic, and it is used if somebody has been exposed or thinks they've been exposed. They can be treated with AZT to prevent the infection from promulgating. And it can also be given to women who are breastfeeding, for example, to limit the risk of transmission to a suckling infant. Things like that. It is a good prophylactic, and it is still used as a treatment in third world countries where they don't have anything else. The protease inhibitors are much, much, much more effective, as I've said, and they prevent the production of mature particles, and people can be treated so successfully that they have no circulating viral RNA at all, and they are considered non-infectious once that happens. The HART is a combination of these various protease inhibitors. It's highly active antiretroviral treatment. It uses several drugs that are rolled over time to prevent the development of resistance to the drugs. And unfortunately, all attempts to produce a vaccine, which are still ongoing, have not resulted in any success whatsoever. I can't finish without mentioning some of the prevention that you need to know about. Unfortunately, information about HIV is no longer as widely disseminated as it used to be, nor, for example, are condoms as easily acquired as they used to be. Many people now have to go to the pharmacy and buy them, whereas beforehand they had bowls of condoms at every bar for people to use. So the very first thing is to avoid really high-risk behavior, like sharing needles. If you are an intravenous drug user, make sure you have your own kit and that you use clean needles, clean syringes, and never, ever, ever share with anybody. The other thing, of course, I've already alluded to, is avoid unprotected insertive sex. And that includes oral sex. The virus can be acquired through oral sex. It is not common, but it can be. So condoms, the simple thing that a condom can prevent acquisition of the virus. That has saved more lives than we can number. This really simple thing for people to use. But the other thing, fortunately, for those of you who are sexually active, and especially if you're sexually active with many partners, you can take what is called PrEP, pre-exposure prophylaxis. This PrEP contains low doses of protease inhibitors, and these prevent infection. Even if you should be exposed to the virus, it prevents infection. And this PrEP is not only for people involved in gay sex, in same-sex interactions. It's for everybody. Anybody who is sexually active and has multiple partners should consider going on PrEP. You can talk to your healthcare provider about it. Or if you're gay, you can go to the Gay Lesbian Community Center in the village and get full information about going on to PrEP, and find medical providers who will give you PrEP to use. It's an excellent, excellent, excellent drug regimen. And it has the so low dose that it has very, very little side effect at all. Okay, so I apologize for this very lengthy recording, but I wanted to get all of the viral disease stuff out of the way so that we can now start to talk about bacterial disease. And I will see you on Monday. Don't forget, you have a test on Monday that is bacterial growth and DNA. The DNA tutorial will serve you well. I've put up the answers. Bacterial growth, I'm just relying on you to structure your own studying. It seems so straightforward that I didn't provide a study scheme. And I will see you on Monday.